All right, so now we're going to move into some general information about different types of microorganisms. So we're still working on kind of a general introduction to microbiology, and we're going to kind of touch on the different types of microorganisms, and then throughout the semester we're going to get into some more details about all of these different types of organisms as we look very specific, or look at very specific examples of each of these. So up here at the top we're at 1.3 types of microorganisms. And most microbes are unicellular and small enough that they require artificial magnification to be seen. So we've already talked about how microorganisms, meaning small, micro, are difficult to see with the naked eye. So we have microscopes that allow us to see those, which is a type of artificial magnification. However, there are some unicellular microbes. So unicellular, again, meaning single. Uni is singular. And cell, so single-celled microbes that are visible to the naked eye, and some multicellular, so many-celled organisms that are microscopic. So there are examples of both, is kind of the point of the sentence there. An object must measure about 100 micrometers to be visible without a microscope. So if you look at this image down here, they show you the area right here where we can use a light microscope to see different things. Down here, these are so small that we need to use an electron microscope. So even the light microscope, like in our labs, for example, are not able to see some of these really small items down here. So it has to be at least 100 micrometers or micrometers to be visible without the microscope. So we're looking at, here's about 100 micrometers. So something larger than this we might be able to see if we're looking really close without a microscope. But most microorganisms are much smaller than that. You can see how the scale is here and how most things are to the left of this. So not many things are to the right. When we're talking about microorganisms, it's mostly smaller than what we can see with the naked eye. For some perspective, it mentions, consider a typical animal cell. So here's an animal cell on the chart here that measures roughly about 10 micrometers. So here's our 10 micrometers. It's kind of in this general area you can see here, about 10 micrometers across, but it's still microscopic. So we're not really able to see individual animal cells. And then you have bacterial cells. Here it mentions bacterial cells are typically about 1 micrometer. So much smaller than that. Down here we have our bacteria cell, typical bacterial cell. Of course there are variations. You know, there are animal cells that are much larger. So if you look at a human egg, it's much larger than, say, a skin cell, for example, or a blood cell, which is all the way down here. And there are different sizes of bacteria as well. So it's just a general size that they put on this chart. And viruses can be 10 times smaller than bacteria. So if we look, here's virus right here, smallpox virus, but even smaller we have the flu virus, and even smaller yet we have the polio virus there. Viruses are generally much, much smaller than bacteria. Bacteria are generally much smaller than animal cells. So then we're going to move down past this chart here. We do have a scale here, or units of length that are commonly used, so we're going to use a lot of these terms. If you remember any math courses, if you've taken some math courses, here's our nanometer, micrometer, millimeter, centimeter, decimeter, and meter. So we're going to be working mostly down in this area, millimeter, micrometer, and nanometer. Maybe sometimes we'll be talking about centimeters, but that's about the area we're going to be working in this course. Microorganisms differ from each other, not only in size, but also in structure. So the structure of the cell, some have a membrane-bound nucleus, some do not. Habitat, where they can live. So for example, a human egg or an animal cell is not able to live in some extreme environments that bacteria or viruses can live in. Metabolism, meaning kind of what it uses to live. So that varies widely, what is used for the function of that organism, and many other characteristics. And again, we'll look at these in detail when we're talking about specific organisms as well. While we typically think of microorganisms as being unicellular, there are also many multicellular organisms that are too small to be seen without a microscope. So some of these, for example, we'll be seeing throughout the semester. So we're going to be taking a look at some larger things. We can look at some yeast cells, some mold cells, and those are pretty large. Some microbes, such as viruses, are even acellular. A meaning without, and then cellular, so without cells, not composed of cells. Microorganisms are found in each of the 3 domains of life, so archaea, bacteria, and eukarya. Microbes within the domains bacteria and archaea are all prokaryotes. So hopefully you remember back to some general biology courses that their cells lack a nucleus. So prokaryotic or prokaryotes do not have a membrane-bound nucleus. So their DNA is kind of floating all over within the inside of the organism. Whereas microbes in the domain eukarya are eukaryotes, and their cells do have a nucleus. So their nucleus is all kind of in one area, and it has a membrane around it, making sure it stays in that area. Some microorganisms, again, such as viruses, do not fall within any of these 3 domains of life. So it is undecided where viruses kind of fit in things, but since they do not have cells, they're not composed of cells, they're not considered living. So they're kind of on their own chart. They have their own thing going. In this section, we will briefly introduce each of these broad groups, and we're going to move into that. And then in later chapters, we're going to go into a lot more detail about each of these specific things. All right, so we're going to start with our prokaryotes here. So prokaryotic microorganisms. First, we're going to talk about bacteria. And bacteria are found in nearly every habitat on Earth, including within and on humans. So most bacteria are harmless or helpful, and we've already talked about that a little bit as far as fermentation is concerned. But some are pathogens. So remember, pathogen means that it causes disease in humans or other animals. So talking about pathogenic bacteria, and then we also have harmless or helpful bacteria. Bacteria are prokaryotic because their DNA is not housed within a true nucleus. That's another term for describing a membrane-bound nucleus is a true nucleus. Because I think that term comes from the fact that a lot of times, going back to giving things specific names that have a genus and species, that sometimes when people would look at bacteria underneath the microscope, they would see that all of the DNA is in one particular area, and it's all very tightly packed sometimes. And so then they would say, oh, well, look, the DNA is in that one little area. It must have a nucleus. But if they looked even closer, they would see that it's not a true nucleus. It would have been like a false nucleus, for example, because it's not bound within a membrane. It's not actually contained in that area. It just happened to all be wrapped up in that particular area. So true nucleus would really mean that it is membrane bound. Most bacteria have cell walls that contain peptidoglycan, and we're going to talk a lot more about that when we get into looking at the very specifics of bacteria cell. Bacteria are often described in terms of their general shape. So the common shapes include coccus, which is a spherical shape. So we're talking about like the round shape, but it's a sphere, of course, because it's 3-dimensional. Rod-shaped, which is also called bacillus. You can see the illustration here, but then also some actual magnification photos or microscope photos. Curved, which can include 3 different types here. Spirillum, which would be this one over here, so it's kind of wiggly. Spirochete, which is over here, which is kind of like a wound up telephone cord, like a really old telephone cord here. And then Vibrio, which basically just means kind of curved or comma-shaped. They have a wide range of metabolic capabilities. So bacteria are, since they live in nearly every place on the entire Earth, they have a wide range of metabolic capabilities. So something that is living in the Antarctic, for example, would have very different capabilities as far as what it kind of eats, metabolizes, compared to something that's inside of our bodies, for example. They can grow in a variety of different environments. So there are some that are called extremophiles, which can live in extremely hot places or extremely cold places, like I just mentioned, in places that are covered in ice, using different combinations of nutrients. So depending on where it's located, depends on what it's going to be using as a nutrient in order to grow and metabolize things. Some bacteria are photosynthetic. So that means that they can use the sun in order to exist, in order to do their metabolic processes. So such as oxygenic cyanobacteria. So cyanobacteria is an example of one that's photosynthetic, as well as anoxygenic. So oxygenic, meaning utilizes oxygen or produces oxygen, cyanobacteria. And anoxygenic means without being oxygenic, green sulfur, and green non-sulfur bacteria. So these bacteria use energy derived from sunlight. So that's the photosynthetic part. So if you remember back to general biology, photosynthesis is derived from sunlight, and then they fix carbon dioxide for growth. Other types of bacteria are non-photosynthetic, so they do not go through the process of photosynthesis, and they obtain their energy from organic or inorganic compounds. And we're going to look at different examples of each of these as we get into the chapter on bacteria, and throughout the entire semester, in fact, because we'll be doing a lot of work with bacteria. Archaea are also unicellular, so single-celled, prokaryotic, meaning they do not have a true nucleus or a membrane-bound nucleus, organisms. So archaea and bacteria have different evolutionary histories. Going back to that tree of life, bacteria kind of goes off on its own at the beginning there, and then both Animalia and Planta, which then later had archaea, is going to be off to the other side. So they have different paths there, different evolutionary histories, as well as significant differences in genetics, metabolic pathways, and the composition of their cell walls and membranes. And again, we're going to look at those very specifically as we go along into different chapters. Unlike most bacteria, the archaeal cell walls do not contain peptidoglycan. So remember when we were just talking about bacteria right up here, we mentioned peptidoglycan, and that's in the cell wall. We don't find that in animal cells, so that's something that's specific to bacteria. And then also in archaea, we don't see the peptidoglycan. So that is something that is specific to bacteria. But their cell walls are often composed of a similar substance called pseudo-peptidoglycan, pseudo meaning fake. So fake peptidoglycan, or something that perhaps seemed similar to peptidoglycan. Like bacteria, archaea are found in nearly every habitat on Earth. So archaea are also able to adapt to their environment. So they're found in extremely cold places and extremely hot places. Or extremely, here's a good example also, very basic or very acidic. Some archaea live in the human body, but none have been shown to be human pathogens. So we know that some bacteria are pathogenic, but we haven't seen that in archaea. So moving down here, this is a photo here from Yellowstone National Park that just show that some archaea live in extreme environments, such as this pool here that's a hot spring. You can even see the heat coming off of it there at Yellowstone National Park. And it's interesting because these color changes in the pool are just because of the different microbes that live there. So as it gets hotter down in the center area down here compared to up here, and has a different composition of the water in there, that is going to give different microbes their kind of space there, and the different colors are for different microbes. All right, so now we're going to move into eukaryotic or microorganisms. So we did the prokaryotes, now we're going to look at the eukaryotes. And first what we'll take a look at are protists. So let me go back up here for a minute. So remember, the domain eukarya includes all eukaryotes. Remember that eukaryotic or eukaryotes have that membrane-bound nucleus. It includes unicellular or multicellular eukaryotes, including, so we have the group protists, fungi, plants, and animals. So if we break down the eukaryotic group, we break it into those parts. And then we're going to go down and break those down a little bit further. So when we talk about protists, they are unicellular eukaryotes. So remember, single-celled, and then eukaryotes, meaning it has a membrane-bound nucleus. And they're not plants, animals, or fungi. So we're just looking at this group at the moment, the protists. And within the protists, we have algae and protozoa. So we have eukaryotic. Within eukaryotic, we have protists. Within protists, we have algae and protozoa. So first we'll take a look at algae. Our singular is alga. Our plant-like protists that can be either unicellular, so single-celled, remember, or multicellular, multiple cells. Their cells are surrounded by cell walls made of cellulose, which is a type of carbohydrate. Algae are photosynthetic organisms, so it can undergo photosynthesis, uses the sun, and they extract their energy from the sun, and they release oxygen and carbohydrates into the environment. Because other organisms can use their waste for energy, the algae are important parts of many ecosystems. So especially when we think of things like the ocean, we think of the algae that's in the ocean. It's kind of the bottom of the food chain there, and it's really important to have that in there because a lot of organisms start the food chain there. They are able to eat the algae because it produces the oxygen and the carbohydrates, or eat all of that, or metabolize those things that are released, and then that allows the organisms to grow from there. Also, many consumer products contain ingredients derived from algae. Remember at the beginning of the chapter we talked about how humans are able to kind of harness the power of these different organisms. We utilize them. And so one of the ways that we use a protist or an algae is using it in ice cream, salad dressing, beverages, lipstick, and toothpaste by way of carrageenan. Perhaps you've heard of that before, carrageenan or alginic acid, which are found in those items. It's used kind of like a thickener. It gives it some thickness to these different items. So to thicken up ice cream or to thicken up salad dressing or beverages, to give some thickness to these items as well. A derivative of algae also plays a prominent role in the microbiology laboratory. Something we'll be working with a lot is agar. And so we use agar, which is a gel that comes from algae, and we mix that with different nutrients to make our petri dishes. And then that's what we'll be growing a lot of microorganisms on. So agar is extremely important for microbiology, not only because it's derived from algae, which is a microorganism, but also because it's what we use to grow lots and lots of other things on it. And as you can see here, we use it in a lot of items that we have on the shelves in the grocery stores. So then moving down here, we're going to start talking about the protozoa. So remember, protozoa is one of the groups of protists. Okay, so we're still talking about protists. We talked about algae, and then now we're going to talk about protozoa. Protozoa. So, singular, protozoan, are protists, again, that make up the backbone of many food webs. So again, we're talking about some of the very bottom or the very basic parts of our food webs, or used to be called food chains, but now it's so interwoven that we call them food webs. And so protozoa and algae, these protists, are kind of make up this very bottom part. And there's a lot of them, which is great because then they can be used further on down the line. So they make up the backbone of food webs. They provide nutrients for other organisms, just like the algae do. Protozoa are very diverse, again, kind of all over the place. They are very, very different from one another. Some protozoa can move with the help of hair-like structures called cilia. If you remember back from basic biology, cilia are little tiny hairs around a cell and allow it to kind of push fluid by it using these little hairs in a motion. It'll lift the hairs up and then push them one direction, and it kind of pushes the fluid down and allows them to kind of shoot forward a little bit, propelling themselves. Or they can use whip-like structures called flagella. And so the flagella oftentimes kind of spins in a circle, and it allows the microorganism to kind of move itself through the fluid or through the area. Others extend part of their cell membrane and cytoplasm to propel themselves forward. These cytoplasmic extensions are called pseudopods, or pseudo meaning fake, and pods for feet, so fake feet or false feet. So what they'll do is they'll kind of glob out a part of them and then suck all of the rest of it into that section that globbed out, and then do the same thing. Like throw another piece of section out there and then move the whole interior part of it, and then throw another piece out there and move the rest of it. So they're fake feet or false feet because they don't actually have feet. They're just kind of lobbing out part of their body and then filling the body or that section in by moving that fluid over there. Some protozoa are photosynthetic. and others feed on organic material, so we have both there. Some are free-living, whereas others are parasitic, where they're only able to survive by extracting nutrients from a host organism. So some can be floating out there freely, getting things from the environment, but some are only able to live within something else and just thrive by soaking up stuff from that one particular host organism. Most protozoa are harmless, but some are pathogens that can cause disease in animals or humans. So one example here's the intestinal protozoan parasite that infects humans and other mammals, and it's Giardia lamblia. This is an example here of Giardia lamblia, which is kind of floating around in waters, and then it can get in through the skin. And then when a person has Giardia, they tend to have severe diarrhea. It can be life-threatening at times, but if it's caught early enough, then it can end up being okay. All right, so our next section we're going to talk about are the fungi. Fungi, or singular is fungus, are also eukaryotes. Remember, we're talking about eukaryotes right now, not prokaryotes. Some multicellular fungi, such as mushrooms, multicellular mushrooms, they resemble plants, but they're actually very different than plants. So remember that plants use photosynthesis in order to provide energy and allow the plant to grow. In this case, one of the main differences between plants and fungi are that they are not photosynthetic. Also, their cell walls are usually made out of chitin rather than cellulose. So cellulose is something that is found in plant cell walls, gives it some structure, and chitin is something that we find in fungus cell walls. And again, they're not photosynthetic. They're unicellular fungus, remember, and multicellular fungus. So an example of a multicellular fungus, again, is mushrooms. And unicellular fungus, an example would be yeasts. They're all included within the study of microbiology. There are more than 1,000 known species, so lots and lots of fungi. Yeasts are found in many different environments, so again, from the deep sea to the human navel. Some yeasts have beneficial uses, so causing bread to rise. We've already talked about how that is very beneficial for humans to be able to make bread. We allow that yeast to sit in there and ferment, and then that rises, or beverages, other beverages to ferment or to make those beverages. But yeast can also cause food to spoil. So when we have yeast, it can spoil the food. Some even cause diseases such as the vaginal yeast infections or oral thrush. So we kind of have all ends of the spectrum there as well. We find yeast very helpful, and we actually package that up and sell that in the grocery store so that you can make bread and other items, to causing food to spoil, which we don't really prefer, and causing some diseases like oral thrush, for example. Here is Candida albicans, and that is the unicellular fungus or yeast, and that is the one that we were just talking about that causes the disease, the vaginal yeast infections, as well as oral thrush, which is basically a yeast infection that can be in the mouth, and that's often found in infants. So other fungi of interest to microbiologists are multicellular organisms called molds. So we're moving back to a multicellular organism. Remember, we were just talking about a unicellular one, and we were talking about the yeast. Now we're talking about multicellular organism. Another fungus is called a mold. So keep in mind that molds are an example of a fungus. They're made up of long filaments that form visible colonies, and they're found in many different environments, again, from the soil to rotting food to our dank bathroom corners. Molds play a critical role. They decompose dead plants and animals, so it's necessary that we have them in order to continue with that whole food web. Some molds can cause allergies. Others can produce disease-causing metabolites called mycotoxins. So mycotoxins can cause severe reactions in a human or an animal. Molds have been used to make pharmaceuticals, so if you think of penicillin, that's a pharmaceutical, which is the most commonly prescribed antibiotic. And cyclosporin, which is used to prevent organ rejection following a transplant. So again, we see the same situation here with molds, where on one end it's decomposing dead things, which is great for the food web. or is causing allergies and mycotoxins that are dangerous to humans. But on the other end, we have things like penicillin or cyclosporin, which is very helpful to humans. And so then here's a picture of some fungus there, some molds on these moldy oranges. Moving to our next section, we have the helminths. So a multicellular parasitic worm is called a helminth. And so maybe you're thinking, how could this possibly be a microorganism? It's very clear that it is not micro. They're not technically microorganisms, but we do study them in microbiology because these worms fall within the field of microbiology because of their eggs and their larvae. So looking at their life cycle, it kind of starts in the eggs and the larva stage inside of a human, and so it is put in the category of microbiology. One example of a helminth is the guinea worm, so Dracunculus medinensis, which causes dizziness, vomiting, diarrhea, painful ulcers on the legs and the feet when that worm is working its way out of the skin. Infection typically occurs after a person drinks drinks a beverage that contains water fleas that have been infected by the guinea worm larva. So the larva actually infects these water fleas, and then the water fleas are in the beverage, and then the person drinks that, and then they get that guinea worm larva into the body, and then that's able to grow and then turn into this large parasitic worm. In the 1980s, about 3.5 million cases of this guinea worm, but mostly that is gone now. In 2014, there were only 126 cases reported, so we don't see that very often, and oftentimes it's in more third world countries rather than the first world countries. And that's due to the WHO, the World Health Organization, as well as other groups that have been trying to get better drinking water, clean drinking water, and better sanitation. So this first one here is actually the Taenia saginata, which is the beef tapeworm. I can do a little smaller thing there, and that infects both cattle and humans. So again, even though this is very large, you can see that this is very clearly seen without a microscope. The eggs are microscopic, and so it's kind of thrown in with the microbiology here. And then B is the adult guinea worm, the Dracunculus medinensis, and this right here is how these worms are removed. They actually take a matchstick oftentimes. Remember, this is related to third world countries, so a lot of times they'll just take a stick, or a matchstick in this case, and then they start to wrap the worm around it and then slowly keep turning that matchstick to just kind of pull the worm out of the skin, slowly so it doesn't break, of course. And then once that's twisted out of the skin, and then the person no longer has that helminth in their body. All right, so moving on to the category of viruses. So remember, viruses are acellular microorganisms. So we're not talking about unicellular or multicellular at this point. We're talking about acellular. A meaning without, and cellular meaning without cells. So they're not composed of cells. Essentially, a virus consists of proteins and genetic material. So a virus can have either DNA or RNA, but they don't ever have both. And they are inert outside of a host organism. So that's another reason why we don't consider viruses as being living organisms, because they're not able to live outside of its host. It can only ever be living inside of a host cell. However, by incorporating themselves into that host cell, viruses are able to co-opt the cellular mechanisms. So a virus gets into the cell of a host, and then it kind of takes the controls. It's a big machine inside of the cell, and it takes over the cell, and it uses the components that are in that cell host to multiply and to infect other cells or infect other hosts. Viruses can infect all types of cells, so from human cells to the cells of other microorganisms. There are bacteriophages, which are viruses that infect bacteria, so you can imagine how teeny tiny those are. In humans, viruses are responsible for numerous diseases, from the common cold to Ebola. However, many viruses do not cause disease. So again, we have everything from one end of the spectrum to the other. So some that can be very, very dangerous to some that are not dangerous at all. All right, so here, just a couple of photos here. A is a coronavirus. So you can see that there. These microscopes are very, very, very small, are able to magnify very, very tiny things. This is a transmission electron microscope, or TEM. B is an Ebola virus, so right here. So that also is using a TEM microscope, or a transmission electron microscope. Microbiology as a field of study. So microbiology, as you can see, is a broad term. So we're talking about everything from an acellular organism to a unicellular to multicellular. We're talking about prokaryotes. We're talking about eukaryotes. So it talks about, or helminths even, which is, ends up being a huge worm, or some are huge worms. It encompasses the study of all different types of microorganisms. But in practice, microbiologists tend to specialize in one of these fields. So there generally isn't a microbiologist that's out there that's studying viruses as well as helminths, for example. Those are two very, very different microorganisms, and people tend to specialize. Here are some specializations. Bacteriology is the study of bacteria. Mycology is the study of fungi. Protozoology, the study of protozoa. Parasitology is the study of helminths and other parasites, so we'll talk about a lot of other parasites. Virology is the study of viruses. So you should know those different categories. You should know the bacteriology is the study of bacteria, and mycology is the study of fungi. All of those different things, you should be able to connect those. Immunology is the study of the immune system, and that is often included in the study of microbiology, and we will include that. That's going to be probably the last chapter, or at least close to the last chapter that we study throughout the semester, because it is so closely tied into microbiology, or microorganisms, because we get to look at that host-pathogen interaction. It's central to our understanding of these infectious disease processes. Microbiologists can also specialize in certain areas of microbiology, such as clinical microbiology. So in the clinic, right, more like a medical microbiology. Environmental microbiology, so looking at microorganisms in the environment, perhaps looking at some of these larvae and the eggs for some of the worms or some of the protists. Applied microbiology or food microbiology. In this textbook, we are primarily concerned with the clinical applications of microbiology. But since the various subfields of microbiology are very interrelated, we'll often discuss these applications as well. So it is understood that the study of microbiology, or that this course of microbiology, is often taken to be used in a clinical setting. But also, it's so closely related to all of these other things. We can talk about it as far as environmental biology, or as far as the food we eat, as we've already done several times. So this is an interesting thing. I recommend reading this bioethics in microbiology. I won't be testing on that or anything, but it's quite interesting. Some of the things that were done when studying microbiology a while ago in the 1940s. So I recommend reading that. That's an interesting little section. So then here's our clinical focus. This is kind of the end of the clinical focus there that kind of wraps things up. And then each of the chapters has this kind of summary, which I recommend looking at. It does a nice job of breaking down each of the sections. So it's nice for a little guide, things to remember, kind of study from, maybe things that you want to put on a flashcard, for example. And then at the end of the chapter, I always recommend doing the review questions. There are multiple choice questions you can see here. There are fill-in-the-blank questions, short answer questions, critical thinking questions. So I definitely recommend doing that. It's important that you really understand this material, and you're responsible for knowing the material on quizzes, exams, in-class work, homework, and those types of things. So if you can answer these questions, then you're just going to do all the better in the other coursework. So I definitely recommend that.